And let me try sharing the screen again. Is it visible?
Yes, ma'am.
Yes, ma'am.
Okay. Okay. So what you're looking at is just by seeing this image, it's a very high speed image of bacterial motion, very dense bacterial motion. You would see that you see some sort of a you see some sort of a circular motion, right? It seems that things are going in circles. So there is a certain directionality to this. Now, I would not go into the physics of it partly because I'm not quite qualified to be teaching those aspects. But I would go right into how these systems are. It's just to try to give you a glimpse of what is being done or what is being thought about when people try to think of futuristic designs with biological motors. So if you now look at, or if you, in this very dense bacterial suspension, if you drop a very tiny particle in it, tiny particle of the order, the size is of the order of the bacteria that's swimming around, and now you try to track this particle. This particle is not going to be active particle because it doesn't have a flagella, it doesn't move by itself. But if the bacteria is indeed moving in a particular direction, in a particular orientation, then this particle would start taking that orientation or taking that direction. So these are called tracer particles. It's almost like you drop a paper boat in a river and you see where the paper boat is going and you know the direction of the river. So it's almost like that. You drop a particle, but the size of the particle should be very close to the size of the particles that is creating the flow, so the bacteria in this case. And then you look at how the particle is behaving. So they did such kind of an experiment, and this is what they see. You look at this black dot and you see the kind of ellipsoid movement, elliptic movement of that black dot. So again, you can see that the tracer is actually creating an elliptic movement. That does not mean that there would not be some traces which are not creating elliptic movements. But it is possible, but it is definitely possible to have a tracer that can very successfully create a very specific geometric movement, and you can, and by that you can understand that the flow, the direction or the flow lines, how the flow is created and how it is moving. You can actually visualize it by putting such trace elements. Okay, so this is one system of bacteria, very dense bacterial suspensions, and you see that a certain pattern of flow is emerging in such dense suspensions. If you try to do the same thing in a school of fish, so when you have these huge number of fish and you try to see exactly how they move, you will see that there is also a flow line there. So if you so what are the kinds of flow that you can think of? You can think of streamline flows, you can think of turbulent flows. So can anybody explain what a streamline flow and turbulent flow is? You must have read it somewhere. At least the words themselves have some, may give you some idea.
Ma'am, a streamline flow is a flow like in which all these fluid particles are flowing parallelly, and turbulent flow is a flow in which all these fluid particles are randomly flowing in random directions, and their velocities are also not the same,
Okay.
and we cannot.
Okay, okay, thank you, Piyush. So to understand whether this is a streamlined flow or a turbulent flow, you would want to not just look at the particle in space and in time. You would also like to you would not just now, you would like to know how these particles behave in space and time, right? You would like to know how they're moving with time. So if you want to look at how they're moving with time, you do something which is called a particle particle image velocimetry, which gives you something like a trace line, how the particles are moving with time. Exactly what you saw with that small black dot, which was moving in an elliptic movement. So it was giving you exactly the way the bacteria is creating the flow. So you could see how the flow was going on in that system. So in dense bacterial suspension, in very dense bacterial suspension, as you keep increasing the number of bacteria in your suspension, you make it more and more dense, you would see that it goes from random, initially bacteria, if you have less number of bacteria, you can see that they would be going in all directions. If you have not given it some kind of a gradient, say a gradient of light or a gradient of nutrients or attractants or repellents, you would see that they would be moving in all possible directions. And as you keep increasing their density, you can see that certain flow patterns emerge. And these flow patterns could be flow in a particular direction, could be some kind of turbulent, vortex-like flows, etc. And so we these are characterized, as I said, the physics behind it is characterized as being very similar to what would be considered a liquid crystal, where you have particles which all have a certain directionality to it. Maybe एक particle ऐसा है, एक ऐसा है, but ultimately the directionality of it would all be in one direction. Okay. Now, why am I saying all of this? I'm saying all of this because you could use these bacteria, you could harness the motion of the bacteria to move certain objects. Now, one example of that has already been given here, where you have taken this dense suspension of bacteria. So you have this dense suspension of bacteria, and if you now were to drop a particle, any particle inside the suspension, you can see that the particle traces a certain path. Right? You can see it traces a certain path. Another way by which you can harness so this is one idea. The other idea is that because bacteria move so fast, what if you design some sort of nano rotor, a nanorotor, and you use the power of bacteria to move this rotor. So this is one such design. You can see that these are rotors which have these grooves in them. And these grooves are of the size of the bacteria that we wish to trap. The idea is that once the bacteria that is moving in solution gets trapped here, then the flagellar movement of the bacteria would start powering the motion of this rotor. And we will have a look at this video to understand how exactly this has been done. So this is the design of the rotor. It's the design of the rotor. You can see the scale of the rotor is in micrometers. And you can see that the size here is less than, say, 5 micrometers, which would be roughly the size of the bacteria that you wish to trap. The idea is that when the bacteria flows, some of them would get trapped in this groove, and that would start powering the motion of these rotors. And in this video, what you see is a number of these rotors which have all been placed. Now, what is interesting is that the way you angle these grooves would also allow you to ensure that the rotor moves in either clockwise direction or counterclockwise direction. So if you were to angle it in the opposite way, you would see that the rotor would move in the opposite direction. Let's now have a look at how these motors move. You see the bacteria that are moving all around, and you see some of them actually go in and they get into these grooves. They sort of stick into these grooves and they remain there, and this rotor is powered by that. So these are channels that are allowing these bacteria to get in. Once they get in, they start attaching to these grooves, and you can see that sort of powers the movement of the rotor. Now the bacteria are made fluorescent because they have red fluorescent protein expressing in them. So if you look under a fluorescent microscope, this is what you can see. The same thing under bright field. If you image it under bright field, this is the way you would see it. But if you put it under a fluorescent microscope, you can very easily see that these grooves are now, the bacteria are now embedded into these grooves and are powering the motion of these rotors. Now there are certain, it's not a very simple design. Rather, they have tried a lot of things. They have even angled this rotor at a certain degree so that this rotation is possible. You can, if you would like to have a little more idea about this entire system, how this has been designed, you can go ahead and look at the paper from which I have cited here before. So this is the paper which shows how this design has been done. You can see that this axis of the rotor is actually kept at a certain angle, and that angle ensures that it is rotating. If it was not angled, then it would not have rotated. I mean that's all explained in this paper. I would not go into those parts, but for us, what is interesting is that these bacteria and the motion of the bacteria can be harnessed at least in these different ways. One is that you can very clearly and specifically use them to very clearly and specifically use them uh, uh, to to propel a particle and to make it follow a certain path and with very high velocities. The velocities here is, as I said, the velocity achieved in these flows is more than the velocity of a single bacterium. If the velocity of a single bacterium is, say, 10 micrometers per second, this can go even high as 20 micrometers per second. So this increased velocities, you can propel very small tiny particles in space and with a very specific geometry at very high speeds, and you can use the motion of bacteria to propel. Oh my goodness. You can use the motion of bacteria to move some rotors in a given direction, but you have to design that rotor in such a way that it can harness the power of bacteria. Now, why would we like to design this, and what can we think can be done with this? Well, it's not entirely clear what we could be doing with this. It's as I said, it's a futuristic design. You could be propelling or powering different things out of it. You could be making tiny robots out of it. But what exactly we could be doing is not very clear right now. Maybe in 10, 15 years it would become more evident what these systems could be used for. But at least right now, it's in a developmental stage to see what are the things that could be done out of it. Now, this is another example that I have picked out to give you a glimpse of what we could do out of these biological motors. So, one of the challenges of working with biological systems or with very tiny microscopic systems is that you would sometimes require ways to purify as well as to concentrate them. Um, for example, let's just say that you are trying to isolate some pathogens that are present in the bloodstream of a patient. And because the pathogen numbers are low, you would need a way to concentrate them before you study it. So there are various ways by which it is done. But here we are going to look at a method where they are not using. So one of the ways by which it can be done, I'll just give you a brief idea, is that you could tag you could use antibodies that bind to this bacterium, and then you could use a method to isolate those antibodies. In the process, when you're isolating the antibodies, you're also isolating the bacterium. That could be based on a number of techniques. You could put a magnetic bead at the end of the antibody, so the antibody binds to the bacteria. Now, if you put this entire system under a magnetic force, these antibodies would stick to the magnet, and therefore you would be concentrating all the bacteria on that magnet, whereas the rest of the fluid would just flow through. Or you could put a certain fluorophore at the end of the antibody, and then every time you did so, and then you let this entire thing flow in a flow channel, and every time you detect that fluorescence, you could make that antibody tagged bacteria move in a certain direction by applying a small electric field, and that would allow you to separate it out. This is exactly what is done in flow sorters, where you detect the fluorescence, then you apply a certain electric field, and you make those antibody tagged cells, bacteria, whatever it might be, move away from the main flow line. Here what they have done is, not by without using such kind of electric or magnetic forces, how can you how could you sort these molecules based on biological motors themselves? So this is a general scheme of design that they have come up with. Again, you have a flow of these molecules. Any molecule, it could be a bacterium or anything. Here, what they have done is they have done it with biotin. So anything that is tagged with biotin could be isolated from this system, could be concentrated. So your analyte is moving in this direction. The direction and force with which your analyte is moving is controlled by flow of buffers that streamlines the flow of your analyte. And then here, they as this analyte flows, it flows into a chamber here, which is coated with microtubules. It's coated with microtubules, and you have kinesin motors that can work on these microtubules. And the kinesin motors are tagged with streptavidin, such that every time there is biotin that is present in the analyte, it binds to the streptavidin, and the streptavidin is bound to the kinesin. So the entire system just simply walks on the microtubules towards this collector end and accumulates at the collector end here. So this is now, if you tag your analyte, the biotinated analyte, with say you tag it with, say, any fluorescence, say TFP or RFP or anything, you tag it with a fluorescence and look under a fluorescence microscope. You look at this entire system under a fluorescence microscope. You can see the analyte that is moving in this direction. You can see the fluorescence emitted. And as the analyte keeps moving, and as the capturing on the kinesins take place, the kinesins walk on the microtubules towards the collector end, and you see that at the collector end you can see increased fluorescence intensities, which indicates that indeed this system could be used as a method to concentrate your analyte. It could be any analyte, as long as you have a good pair with the kinesin motors. You take your kinesin motors, you put anything on the kinesin motor that can have a complementary pair on your analyte, and therefore capture the analyte as the kinesin motors walk on microtubules. It captures it and it moves it towards the collector. So again, this is again a futuristic design. It's not something that is commercially done yet, but this was kind of a way to show that this is possible. Now, biological systems have their own limitations. The limitations being that they absolutely must require water. So both the systems that I have shown today, everything is happening in water. So you cannot get rid of that. You have water and they need to be powered by some source. So in the first experiment, in the first design that I have shown you, here, you see it is called light controlled. Why light controlled? Because if you are to do such an experiment in micro scales, the idea is to have the bacteria moving for a very long time, and that time is not sufficient enough for the oxygen tension to remain high inside that system. So you need a different way to power these bacteria if the bacteria are aerobic or even facultative anaerobes. Even then, you would need them to have a certain oxygen tension inside the media. That becomes a limitation in a number of cases. So to overcome that, what people have done is instead of you might say use an anaerobe, but very often you might see that your aerobic bacteria are maybe the easiest source available, or maybe they are non-toxic, because that's one of the criteria. We shouldn't have bacteria which is toxic to us, and maybe that's the bacteria in which you have the certain mutations that allow it to move very fast. So it's better that you use that. So to overcome this limitation of oxygen concentration or oxygen tension in the media as you keep moving this system for prolonged periods of time, they have added certain proteins that can harness light energy to actually create ATP, or to create those proton gradients that can drive ATP. So uh, that's the reason why in this paper you would see it's a light driven system, because the light is actually creating ATP. It's becoming the source for ATP synthesis. Uh, so in this system, whereas in this system, you see that when the kinesins are moving, they have an absolute dependency on ATP. They would need ATP for their movement. So when you are working with a system, or when they were working with the system, they had to keep the ATP levels high enough for the kinesins to keep walking on the microtubules for a prolonged period of time. That means you need to replenish the buffers continuously as you keep the system working. So the idea that the biological systems can be used to power some sort of futuristic designs in motors, or to actually use them as small robots that would perform a certain task is not completely sci-fi. It is possible. There are limitations in our own design, and the limitations come from the way these motors work, chiefly because they require water, and they would require some sort of a source of energy. So you would have to continuously keep them in a water system. You would have to provide them with that source of energy for these biological motors to power themselves. And you can find different ways to power these motors, or different systems can be developed to power these motors. Okay, so with that, I will actually stop today's class. It's been a very brief class today. I'm going to stop it now.